We've really come a long way in this course and we've learned quite a lot. And I think it's time for another case study. Now we're going to take something that was published, a paper, and we're going to take a closer look at the research question. We're going to try and identify the study type, what type of sampling was used to get subjects to analyze, what variables were collected to answer our research question, and what data types these variables were, and what the analysis was. So here's our paper. It's the prevalence and characteristics of hepatitis B virus co-infection amongst HIV-positive women in South Africa and Botswana. You see there from Matthews et al., and it was published right this year, 2015, in PLOS One. You see the reference there, read it, quite good information in this paper. Now let's start with the research question. Now a bit of background. Now, the HIV is really endemic in many sub-Saharan populations, unfortunately. But with the progressive availability and success of antiretroviral therapy, that's therapy for HIV, it really has reduced opportunistic infections and malignancy. And those were really main causes of mortality in these patients. So now we have increased survival, and we now see the emergence of previously unrecognized chronic liver disease, and of special concern is viral hepatitis. So these patients live a longer life now. The HIV is under control, and now we see other problems apart from the opportunistic infections and malignancy. Now, there's also an increasing evidence that co-infection, that is HIV with viral hepatitis, specifically hepatitis B, is associated with morbidity and mortality exceeding that as with either of them alone. So there certainly is some evidence of that. Also, many in Africa are vulnerable to liver disease. Now, that's due to genetic factors, that is due to the diet, and especially exposure to toxins, making their livers really vulnerable to infection. Now, there has been some articles in the literature, but the burden and characteristics of this co-infection, HIV and hepatitis B, is only known through small studies of disparate populations. So really the aim of this piece of research was to investigate the burden and the characteristics of the co-infection. So it's really epidemiology and nature. And from the paper itself, they state they aim to assimilate more information about the nature of hepatitis B virus, HIV co-infection in this region. So that was really their research question. So what study type? I think that's an easy question to answer. First of all, it was observational. So there was no intervention. This is not a trial. This is not a clinical trial. It's an observational study. And we want to know how many people are infected, and we want to describe them. So clearly this is a cross-sectional observational study. Let's look at the sampling, and really, if you read it carefully, there's a bit of a mix of sampling techniques. First of all, there was clustering by clinics. So they went to certain clinics, identified certain clinics. Now these are antenatal clinics and pediatric clinics in Durban, in Kimberley. These are a city and a very large town in South Africa, and also a clinic in Botswana. So they looked at everyone in those clinics. So that is really clustering by clinics. And then they stratified patients there according to their HIV status. So they're either positive or negative. So it's really clustering by clinics and then stratification by HIV status. So the HIV positive patients really came from 2 sets of patients in South Africa and then 1 clinic in Botswana. And then also identified another clinic in Durban, South Africa, where they identified 72 HIV negative patients and included them just so that we can compare the HIV positive and negative patients. So in the end, they had a cohort of 1,022 women. Now, if you read the paper carefully, most of these patients form part of previous studies, previous observational studies, certainly a group that has been very active. So let's look at the variables and their data types. So we have a research question, we have this cross-sectional study, and we want better information about the prevalence of this co-infection. So the first was these cohorts, what clinics they come from. If you come from one clinic, the next clinic, or the clinic in Botswana, these are nominal categorical data types. Durban, Kimberley. If we look at the country as well, so it was South Africa and Botswana. Those are not numerical values, even though we can count how many patients come from each. The word Botswana and the data type Botswana and the data type of South Africa, those are nominal categorical data types. HIV status as well, it's positive or negative. Those actual terms, positive or negative, that is nominal categorical data. Hepatitis B surface antigen. Now, that is an antigen is something that sits on the surface of the virus to which the immune system will react. That is for hepatitis B. And it's either positive or negative in the patient's blood. And that positive or negative, that's also nominal categorical data types. Now, the HIV viral load, that's where we actually count the number of viruses. Clearly, that is a numerical data type. And there can be 0 viruses, so there's an absolute 0, so this is a ratio type numerical. And if we look at the numbers involved in this viral load, we really have to see this as a continuous data type, not as a discrete data type. CD4 count, that is a type of immune cell that is affected by HIV in human blood, and again, it has an absolute 0 value for sure, and therefore has to be seen as a ratio type numerical data. But the sheer number of cells that you can count in each individual really means that this is also a continuous data type and not a discrete data type. Now, they've looked at some other variables as well in this article, but some of them become really technical and I don't want to go into them. They really are quite specialist. These are the ones we are going to concentrate on. So let's look at the data analysis. We're going to look at the sections of the results where they looked at the HIV status. So looking at the CD4 T cell count, looking at the viral load, and then also the section on the hepatitis B surface antigen prevalence, so how many patients were also infected with hepatitis B, and then the impact of co-infection, specifically on the CD4 count and the viral count. Let's have a look. Let's start with this HIV status. So in the total of 1,022 patients, you'll note that 950 were positive, so those were from the clinics that they had data on before, and then 72 HIV negative from a different clinic where they just identified the patients that are HIV negative and wanted to include them in this observational cross-sectional study as well. Now let's look at the CD4 T cell count. Remember, that's a type of immune cell in the body attacked by the human immunodeficiency virus. And we see 3 graphs at the bottom that you will not see in the paper itself. Now, the beauty about this, with this paper and why it was chosen for inclusion in our discussion here is the fact that the actual data values in spreadsheet format is also available from the PLOS ONE website. You can download this and look at the data yourself. So these are the 3 cohorts of HIV positive patients. On the left-hand side is everyone from Durban. In the middle, you'll see the patients from data values for the patients from Kimberley, and on the right from the clinic in Botswana. Now, these are probability plots, also called QQ plots, where you take every individual, now these CD4 counts versus its quantile. And you see the little red lines there. Remember, we've discussed this before. If the underlying population parameter was normally distributed. So if we look at the population, we look at CD4 count, now that is the population from which these individuals were taken, clearly these values are not from a normal distribution. And what does that tell us? Well, we can clearly visually see it here because these blue dots do not follow these red lines too well. And if we want to compare these 3 sets of values, we really cannot make use of a parametric test. And indeed, as in this article, they made use of a non-parametric test. Remember also that these are 3 sets of data values that we are comparing to each other. So we have to use the non-parametric version, or non-parametric example of something that would be akin to the ANOVA, one-way ANOVA test. So indeed, as is discussed right at the end of the paper, they used the Kroskal-Wallis test, which is absolutely correct indeed. Now we look at the viral loads for these 3 positive cohorts of positive patients. And again, you'll see you cannot use a parametric test. You cannot use ANOVA to analyze or compare these 3 sets of values, because clearly the data points do not come from a normally distributed patient parameters. Really, we can't do that. And again, they made use of the non-parametric example, and that was the Kruskal-Wallis test, and you can look at the results there. Let's go to the hepatitis B surface antigen prevalence, and you'll see on the right-hand side there that was a contingency table. So of the total of these patients, 950 were hepatitis surface antigen negative, and 72 were hepatitis surface antigen positive. So these are now positive patients and the negative patients. So you can see we can make these, this is a 2x2 contingency table. So you see the column of HIV negative and the column of HIV positive patients, and on the rows, so for each HIV negative and positive, we'll see the numbers that were hepatitis B surface antigen negative and those that were hepatitis B surface antigen positive. Now, they made use of the Fisher's exact test. We've looked at that. They need not have. If we were to do some analysis and we looked at this, certainly the numbers that are in the expected table all would be larger than 5, so we might as well have made use of the chi-square test as well. They decided on the Fisher's exact test, which would also be correct. So this is categorical data, HIV positive, HIV negative. So that's nominal, categorical data, same for hepatitis B surface antigen. So we really are just looking at proportions here. So we use a statistical test for categorical data, either Fisher's exact test here for this contingency table, but as I say, we could use the chi-square as well. Now let's skip to the section on the co-infection impact. Now the authors really compared just the HIV positive patients in South Africa, and they looked at those that were HIV, they made 2 groups from those. So those were hepatitis B viral negative and hepatitis B viral positive. And we're looking at their CD4 counts and their viral load. So first of all, these are the CD4 counts. So these are only patients from Durban. They're all HIV positive patients, and we're just looking at 2 groups here. The hepatitis B negative and the hepatitis B positive. Negative on the left, positive on the right. Again, these are probability plots, not from the graph. You can take the data that they have provided and you can construct these yourself with proper software. And yet again, we see with the CD4 counts, these are not normally distributed data points. They do not follow those red lines. So certainly comparing these 2, we would not use a t-test at all. And if we look at the case for the viral loads as well, again, it's not from a normal distribution in the underlying population as far as the underlying population parameter is concerned. And we have to agree with comparing these 2 sets of data. We have to compare with what the authors did, and indeed they used the Mann-Whitney U-test quite correctly. So wonderful paper here. You can play around with the data yourself, and really such important data, because really the antiretroviral drugs are really changing the landscape, improving the lives of these patients. And as the management of these patients go forward, we really have to address all the issues that come up. And certainly hepatitis B co-infection in these patients can be of concern, and it was very good to have this data from which to work. So wonderful case study. Go and read it. Good paper.